Thank you, Balu, for that nice introduction. It's always an honor to be introduced by one of your former teachers. Now, it's a great honor to be taught by Balu when I was a student at Kanpur many years ago. And it's also a particular honor to speak at the 200th anniversary of Presidency, led by Thank you for inviting me and giving me the opportunity to speak in a university that nurtured J.C. Bose, to my mind, one of India's foremost scientists and perhaps the first neuroscientist, though he was not known as a neuroscientist, and the first polymath who spanned so many different fields. He's been an inspiration for me. Presidency nurtured Mriganka Sur, P.C. Ray, Satyen Bose, so it's a great honor. I must also say that was a long time ago. And so I look forward to Presidency producing the next generation of Indian scientists and shaping Indian science. I'm going to talk today about perhaps the leading edge of science. The mandate I got was to speak on the future of science, its link with the humanities, and how that might help us understand complexity. And perhaps there is no more complex machine in the universe than the human brain, which creates the human mind. And I'm going to take you with me on a journey that takes us to the frontier of this science. How do we understand the brain and how it creates the mind? And takes us, I believe, to the frontier of technology. How might this information help us create the next generation of computation and of computers? And then perhaps we should reflect a little bit based on what we have heard. Sorry. We'll reflect a little bit on not only technological transformation, but perhaps on the issues of social transformation that must accompany technological transformation so that the advances are accessible in a manner that Mamin Chandri has just described. So, most of us know something about the brain. I think our students know at least where it is, and all of us use it. But the human brain is the most complex machine in the universe. It produces the understanding of complexity itself. What can be more complex than that? And it has nearly 100 billion cells whose activity creates the mind. We must agree on that, otherwise it's difficult for a neuroscientist to proceed. And indeed, there is a stream of Indian philosophical thought, particularly the Charvak school, that has argued very articulately for the fact that mental activity is the product of a physical mind. We start with that, and we will ask, how does the brain create the mind? I like to use the word cognition. Cognition as mental activity underlying perceiving, understanding, learning, remembering, communicating, moving in a goal-oriented way, all of the stuff that minds do, but cognition is a more operational, definable term. How does the brain create cognition? Most of what I'll say is about that first question, and then we'll go on to ask, how might this knowledge affect society? And I'll touch upon two ways in which deepening our understanding of the brain and how it creates the mind will affect society. First, in the way that we understand brain disorders and diseases and how we treat them. These are some of the hardest disorders and diseases that we have, because it's about the mind, and you can't see the mind, you can't touch the mind, yet it exists. And second, what I'm going to say, I believe, has deep implications for understanding where next do we go with computers and computation. You heard Mamin say that Tim Cook's salary has been dropped by 10%. Actually, it's 15%. And that's because the iPhone that Apple built in going from a version 6 to a version 7, if you care about these things at all, and it's completely understandable if you don't, there is very little technological advance. The technology of computers, of computation, perhaps even of machine learning that I'll tell you about by the end of my talk, is beginning to hit a wall because the technology of transistors, such as defined by Moore's law, who said every 2 years the density of transistors on a chip will double, that has hit a wall. There are limits, there are physical limits to how many things you can cram into a square millimeter of space. So when the technology that exists today has hit a wall, companies like Apple or other companies that build the next generation of machines have hit a wall unless you transform that technology. And that's where neuroscience and cognitive science come in. And of course, it's an interesting and very deep question to ask, along with this technological transformation, what kind of social transformation would we need, should we need, such that not only can technology be accessible to everyone in society, but what kind of technological transformation does a society want? Okay, so just to take you with me right into the trenches right away. Here are 4 pictures. These are 4 self-portraits by a German artist named Anton Rederscheit. And Rederscheit had a stroke. And he drew these 4 self-portraits. I hope everybody can see them. I'm not quite sure how visible the screen is at the back of the room. Raise your hand if you can see everything that I'm talking about. Because otherwise, thank you, that's great. So I can see hands at the back of the room, so I can assume everybody can see at least the big picture view of what I'm saying. So Rederscheit had a stroke, and a stroke is where a part of the brain is deprived of oxygen. And that deprived of oxygen because the blood vessel that supplies the brain has either gotten occluded due to a blood clot, that's called an ischemic stroke, or the blood vessel has hemorrhaged, and that's called a hemorrhagic stroke. Regardless, neurons or brain cells need oxygen, and if they don't get that oxygen for even a few minutes, they die. Brain cells are some of the most active cells in the body. So then Rederscheit has a stroke, and he looks at a mirror, and he draws a picture of himself. And then a few months later, he draws another picture. And a few months later, he draws another picture, and then another one. A trained neurologist can take one look at these pictures, and tell you where his stroke was. And that tells you something immediately very profound about the nature of cognition. That cognition arises from an internal representation of the world and involves specific networks and dynamics. Rederscheid's stroke was in a part of the brain called the right parietal cortex. It's right here. And as I lose more and more hair, my right parietal cortex, or at least the skin on my right parietal cortex, is more and more visible. And so why does this lead? What is it about these pictures that tell us that's where the stroke is? It's because Rederscheit looks in a mirror and he draws one eye, half a nose, and half a mouth. The left side of his face, I hope you can see it, is entirely blank. There is nothing wrong with Rederscheid's eyes. There is nothing wrong with his ability to draw. There is something profoundly wrong with his ability to pay attention or include in his sense of what an object is anything to the left of that object. So this is what a right parietal syndrome is. It's called left spatial neglect, where it's nothing to do with moving the eyes. He can move his head this way or he can move his head that way, but it's object-centered. In this object, anything to the left of the midline is ignored. It's not attended to. There is no mental construct in Rederscheid's brain that exists. We build internal models of the world when we interpret the world, and these internal models underlie cognition. And this internal model lives in brain regions and is due to activity in brain regions. To understand this, so I'm already taking you into a journey where we are beginning to take apart this very hard question. What is the mind? That you cannot see or touch or feel. Yet the mind has physical consequences. And we can understand what the mind is by understanding the consequences of cognition and by then understanding the brain basis of that cognition. So the brain basis of that cognition lives in neurons. And the brain has 80 to 100 billion with the B neurons. What is a neuron? A neuron is a brain cell. Each brain cell is about 1 to 10, 20 microns. How much is 10 microns? It's 1/10 the width of a human hair. So it's got to be small so that it's crammed into a volume of about 1 liter, which is what our brains are. And there's 80 billion of them, each with these wispy dendrites where inputs come in, and neurons connect with other neurons, and there are 2 ways in which a neuron differs from any other cell in the body, such as in the liver or in the skin. A neuron fires action potentials. It keeps out sodium ions, it keeps in potassium ions, and when sodium rushes in and potassium rushes out, down their concentration gradient, there is a beautifully described biophysics of how neurons generate action potentials. And second, neurons connect to other neurons at specialized junctions called synapses. A synapse is where one neuron ends and another neuron begins. And when a neuron ends, that action potential also ends because it cannot jump the gap. So then that electrical transmission gives rise to chemical transmission. These chemical transmitters are taken up by the next cell, which then change their conformation, and then there is another action potential in the next neuron. It's more complicated than it needs to be, but we carry within us the seeds of our evolution. And it is these seeds of our evolution that form the history of cognition. We can record the action potentials that a neuron makes by many means. For 50 years, we have stuck a wire into the brain and put that wire next to a neuron, and that helps us record these spikes. A spike is a very efficient way of transmitting information. It leads to less losses. It's a beautiful engineering solution, the problem of how do you communicate across many, many different cells? How do you communicate information? But it immediately gives rise to the problem. What is the information that is encoded in the spikes? Neurons fire action potentials or spikes. These spikes have historically been recorded by putting an electrode in the brain, and these spikes encode information in the interval between them. There is no information in the height of the spikes. All of the information is in the interval between spikes. That has been the historic way by which we have understood information transmission and coding. And that's what brains do. Brains encode, transmit, and decode information. And that's one profound way to understand the brain and how it gives rise to the mind. But then, just in the last 10 or 20 years, new technologies have transformed our way of understanding the brain. We know more about the brain in the last 20 years than we have known in all of human history before that. These technologies rely importantly on the ability to image the brain. So you've heard of functional magnetic resonance imaging, where you can put the head in a magnetic field and you can probe the spin resonance of water. One can use population imaging technologies where the availability of oxygen or the activity of large numbers of neurons can be measured using optical tools. One can use multiphoton microscopy to image single neurons or their processes, and one can even image single molecules inside single synapses or the connection or single neurons. That's 5 orders of magnitude of imaging. And like I said, this is the scale of a human hair. This is the scale 1/10 of a human hair. This is the scale 1/100th of a human hair, or 1,000, sorry. So this is an extraordinary range of technologies that can be brought to bear on understanding the human brain, or at least the animal brain. And what have these technologies told us? Well, here's one thing that they've told us, that networks of brain areas underlie cognition. If I were to show you the word car written on a piece of paper, or show you a picture of a car, or show you an actual car, your visual area would light up. If I read out the word car, your auditory cortex would light up. If I were to ask you to speak the word car, your motor cortex would light up. But here's something interesting. If I were to show you the picture of a car, or an actual car, or even the word car, and asked you what verb, what action word goes with this picture or noun, most of you would say drive. It doesn't matter what you say. But 2 other areas light up, and these are the language areas of the brain, one of which creates the concept of speech, and the other creates the motor program of speech. But the important point is this. It doesn't matter whether I show you the word car written on a piece of paper or an actual car. Your output is the same, and that's because human cognition derives meaning from the world in the sense of symbolic representations. We think of a car whether we read the word or we see the word or we are told the word. And this is an internal model of the word. And so I've already taken you down from mind to cognition to an internal model which underlies cognition. And now we are getting somewhere with respect to finding an operational basis for how might this arise. So then, when I say, how does the brain create the mind? I have two questions that I can ask. How is the brain wired? Because it is the wiring of these areas that ends up creating the networks that underlie the mind. And how does this brain wiring then lead to the substrates of the mind, such as cognition? How might we understand these questions? We need systems. And the system that we work on, and that a great deal of our field works on, is the visual system. Vision starts with the eyes, but is really created in the brain. About half of our brain, half of the human brain, or the brains of monkeys that live in daylight, they are called diurnal monkeys. Half of their brain is devoted directly or indirectly to vision and to visual cognition, because vision is so important for us. And vision starts with the eyes, but the eyes tell the brain a set of pixels. And from these pixels, the brain creates vision. And it does so by series of transformations of those pixels into the elements of vision, beginning with the primary visual cortex at the back of the eye, at the back of the brain, to a series of cortical areas that create forms, and other series of cortical areas that create internal dynamics related to vision. So visual cognition arises from a distributed, hierarchical set of networks from the primary to higher cortical areas. And one of the elements of vision that was discovered by two scientists, David Hubel and Torsten Wiesel, in the 1960s, for which they were given a Nobel Prize in 1981, was one of the most simplest of results, which is that when they recorded from the primary visual cortex of monkeys or cats or what have you, they found neurons that did not respond to little pixels of light or to light everywhere, but to an oriented light and dark edge. And you might ask, why is this interesting? Because it was the first demonstration of something very profound, that our cognition has underlying it the responses of neurons in our brain. Is because all objects in the world, you can show this mathematically, I can derive what are called Gabor functions by taking this image and transforming it through a series of filters and Fourier transforms, and then I can reveal to you that the core set of basis functions, what defines any object, is a set of oriented line segments. When you look at me, my face is bounded by little line segments. So in order to put together the image of my face in your brain, there have to be neurons that respond to the little elementary edges. And that's what Hubel and Wiesel discovered. Now, orientation selectivity, therefore, is a key property of vision and is generated within the primary visual cortex from the pixels that the eye provides the brain. And here are two cells recorded in the visual cortex of a ferret by my first graduate student, Anna Rowe, that shows what this orientation selectivity looks like. These are neurons that respond. This neuron responds best to a line that is at about 10 o'clock, 4 o'clock, and moving up to the right. And this is the neuron that likes best to a line at about 1 o'clock, 7 o'clock, moving up and to the left. And it responds much less to any other orientation. So this is what orientation selectivity is. And we can image orientation selectivity. I hope you can see these movies, but we are very proud of these movies where, using very advanced technologies of optical recording, by loading cells with calcium indicators, so that when a cell fires, calcium enters and the cell's fluorescence changes, and by moving a laser at very high speed within the surface of the brain, we can, or looking deep into the brain, in the cortex, in the visual cortex, we can image the activity of hundreds of neurons at a time. And what this tells you is that when this animal, a ferret, is looking at a screen and is looking at different orientations as shown here. Neurons over here respond to one orientation, so verticals. Neurons here respond to those. When the ferret is shown horizontals, neurons here respond. When the ferret is shown obliques, neurons here respond, and so forth. And it shows you two things. It shows you that a neuron here likes one orientation and no other, or a small range of orientations. And second, it tells you that neurons with different orientation preference are not randomly distributed. They are organized as spokes on a wheel. And that's what is called a pinwheel. And if you look at many other neurons across a large area of the cortex, you can see these pinwheels, and a pinwheel is a particularly compact and elegant solution. It's a mathematically elegant solution to the problem of representing all orientations that look out on a region of space within a local region of the cortex. Because a neuron likes one, we see all orientations wherever we look. Therefore, any region of space has to be mapped within the cortex in the context of all orientations. And a pinwheel is the solution. We know this solution mathematically, and nature has discovered this solution by virtue of evolution. So then, we can ask, when we ask, how does the brain get wired? We can use this orientation selectivity as an example. So always we need particular examples in order to learn general principles. How do orientation networks form? So brain wiring relies on genes, molecules, and activity. You have young neuroscientists at Presidency who are studying the issues of how does the brain get wired under the influence of specific genes and the molecules they express. But then, and indeed, orientation selectivity is present at birth, but then it's sharpened by visual experience. There is no such thing as a gene that acts completely independent of an environment. We have the genes we do because of evolution, because of all the ways in which our brain has been shaped by the world we have evolved in. So orientation tuning comes with a genetic propensity to be wired, but then visual experience, the world we grow up in, sharpens that tuning profoundly. And we can ask, how important is this visual experience by rewiring the brain? Now, rewiring the brain helps us understand rules of network development. So in the normal brain, the eye projects to this visual thalamus, which projects to the visual cortex, and the ears, which transduce sound, project through various structures to the auditory thalamus, or the MGN, which projects to the auditory cortex. And very early on in my career, some 30 years ago, I asked this question: what is intrinsically visual about the visual cortex? Can we change the nature of inputs that a cortical area gets and rewire that cortex as a particular example of the importance of the role of experience or of sensory activity in shaping cortical areas and networks? So we took newborn ferrets. And why a ferret? This is a picture. It's a very famous painting by Leonardo da Vinci. It's called Portrait of a Lady with Ferret. And that's a ferret. And you can see at a glance why a ferret is a wonderful animal for visual research. It has frontally placed eyes, just like people do. And importantly, a ferret is born very early in development, at about the stage of the first trimester of birth in humans, so that you have access to these very early developmental events governed by intrinsic factors, and you can alter them. So we took newborn ferrets and we removed the inputs from the auditory midbrain to the auditory thalamus. That changes the molecular architecture of the auditory thalamus, allowing visual fibers from the eye to enter and make contact with the auditory thalamus. But the consequences of this contact are that now there is visual input that begins to drive the auditory cortex. So now we have a cortex that was never made to see, but now gets visual input. And the immediate question to ask is, what does this cortex do with this visual input? And the answer is that the cortex makes orientation selective cells. The auditory cortex makes orientation selective cells. So here are neurons in the auditory cortex, and these neurons have orientation selectivity, and they make pinwheels. So just like neurons in the visual cortex do. The pinwheels are not as elegant, they are not as numerous, but underlying those pinwheels is a profound change in the nature of the circuitry, where in the normal auditory cortex, if you look at which neurons connect to which other neurons, those neurons lie along what is called a sound frequency axis. In the visual cortex of a normal animal, the neurons that connect to each other all lie along the same orientation axis, but are displaced in space. These are just principles of wiring based on ideas of Hebbian wiring, or neurons that fire together, wire together. And in the auditory cortex of rewired animals, the connectivity resembles much more the connectivity of visual cortex than the connectivity of the auditory cortex. So I remind you, this is a cortex that was never made to see. But now when we make it see, it changes its connectivity to process that vision. And this is how we think this is one of the remarkable ways in which the nature of the world, refracted into our brain through electrical activity, shapes our brains in order to process that world. How does this happen? This is another great question in our field. How is it that electrical activity, because that's all that the brain knows about the outside world. The brain has no direct access to the outside world except in the electric activity that is generated through the eye or through the cochlea and reverberates in our brain as spikes. So what is it about these spikes that makes the auditory cortex or any area wire itself? And so it turns out that there are lots of molecules that are involved in wiring within a synapse, and these molecules are regulated across many different time scales by synaptic and electrical activity. And that's another great discovery of modern biology, if not modern neuroscience, is activity-dependent gene expression. And so Onime studies this, and many people study this, and we study it too. And it shows you the deep link between nature and nurture, that it is not nature versus nurture, but nature and nurture together that define brain wiring and connections. So when we look at how electrical activity changes synaptic molecules and synapse function, we find over from tens of milliseconds to hundreds of seconds to hours and days, different molecules change such that they allow different sets of partners to be brought in and wire with different sets of postsynaptic elements. And just as an example, not only during development, but frankly, even ongoing activity changes synapse function and structure. Here's a neuron that we imaged in a living, behaving mouse. And by using 2-photon microscopy, each of these nubbins is a synapse on this one neuron that was labeled using a genetic trick. And when you look at this neuron, you can see, if you image this repeatedly, and I've speeded this up about 50 times, you can see that these synapses are not stable. They are continuously changing. The larger a synapse is, the less it changes. But the smaller it is, the more it changes. So I must have seen 10,000 synapses imaged in this fashion through hundreds of experiments. I guarantee you there are synapses in your brain that are twitching at this very minute. And that's because there are active spines are stable, but inactive spines twitch more and will be lost. And that's how activity shapes circuitry. We can rewire ferrets. We can rewire mice. We can show these principles. And human beings are rewired too. In most extreme cases, human beings born with congenital blindness or with congenital deafness have their appropriate cortical areas not just lying there silently, but they get taken over. They get taken over by other modalities in a manner that enhances the actual processing done by these modalities. For instance, here are the visual areas in congenitally blind humans. And these visual areas are activated by sound localization stimuli or by sound stimuli. In other experiments, others have shown that language, or at least heard language, activates, and the ability to generate speech activates the visual cortex in congenitally blind people. In congenitally deaf people, comparable to our experiments with removing auditory inputs into the ferret's brain or into a mouse's brain, visual inputs end up activating the auditory areas. So then, what I've told you so far is that brain circuits are wired by molecules. These molecules are made by genes, and these genes and molecules are influenced by input activity and by experience. So brain networks are sharing information from the outside world. But then, the brain does not simply mirror the external world. We know that from looking at Anton Rederscheid. His eyes are fine. The eyes are bringing information from the world. But the brain creates an internal model of the world, which is the mind. How might we study the components of this internal model? And that's a great frontier in our field, and I'll give you a little glimpse of it. So, disruptions of cognition reveals the brain's internal model. Anton Rederscheid is not the only person with this right parietal cortex lesion. There are many such people. It's actually a very, sadly, a very common area, relatively common area for a stroke, because this artery called the middle cerebral artery comes up and has its terminations in that part of the brain, and it gets clogged very frequently. So then, here are drawings from patients who have this right parietal syndrome. And remember, they have left spatial neglect, because each side of the brain processes the other side of the world. So when a patient is asked to draw a clock face, this patient only draws half the clock. When a patient is asked to draw a house, he or she only draws half the house or only half the flower. The patient ignores, does not attend to the opposite world. So we know from these experiments and from many other observations that attention filters and focuses information from the world. It's a deep internal state that helps construct the internal model of the world. It's a key component of cognition, attention, and the parietal cortex is a crucial node for attention. All of this we know from this. How might we study this? Well, now I'm going to take you really deep into our field, and then we'll emerge in a few minutes and reflect on what it all means. So I've been telling you about humans, and I've been telling you about ferrets, which are like cats, and now I'm going to tell you about mice. Because we can do experiments in mice to probe cognition. And why mice? Is because mice are small, mice can be trained, mice have cognition. Let no one tell you that animals don't have cognition. They do. They may not have the higher cognition of self-reflection, etc., that human beings have, but they have the components of cognition, and I'll show you that in a minute. So we take mice, but the mouse brain is not just a miniature version of the human brain. If you were to blow up the mouse brain, so a mouse brain is 1,000th the volume of a human brain. It has 50 million neurons as opposed to 80 to 100 billion with a B that the human brain has. And if you blow up a mouse brain, these colored areas are the sensory areas. Here is the visual cortex. Here is the sense of touch, or the somatosensory cortex, or the auditory cortex. The non-colored areas are the higher areas. There is a lot of it in humans. There is a little bit of it in mice, but the little bit there is important and evolutionarily conserved. And so they are called homologous areas. And one of these areas is the parietal cortex. So we can ask, what does the mouse's parietal cortex, the same area that had a stroke in Rederscheid, and that I just showed you the patient drawings from, what do these areas in mice do? And just to set the stage, in order to think about cognitive tasks, here's one kind of task that we do every day when we are driving. We look at a traffic light, and we have learned red means stop, green means go. We see a light, if it's foggy or if it's a little bit smoggy, as it is in Calcutta a lot, then from that sensory evidence, particularly if it's uncertain sensory evidence, we have to make an internal decision. Was it red or was it green? And then, if it's green, then we go. Okay? This is the kind of task that we can teach a mouse. So, we are like circus trainers. By we, I mean my students. If we can take a mouse, and here's a mouse. I promise that I'll take you deep into the trenches with me, and I would hate for you to go away without knowing how do we know what we know. How do scientists know what they say they know? It's crucial, I think, for an educated citizenry to understand that, to understand the nature of evidence. That's the reason why I'm giving you tasks that have uncertain evidence, the nature of evidence, and how does that tell us, how does that advance the state of our knowledge. Here's a mouse. Here's the lick spout. Here's the screen. If we teach this mouse, if it sees a horizontal set of bars, it should lick when this target comes, when this lick spout comes forward. If it sees a vertical set of bars, it should not lick. Okay? That's the task of the mouse. It's as if red means stop, green means go. And we give the mouse a visual stimulus for a couple of seconds, and then we bring the spout forward, and if it's the right stimulus, the mouse has to lick. And the mouse has, this is a 2x2 contingency. The mouse licks when it sees one stimulus. It should not lick when it sees another stimulus. That's called the hit. This is called the correct reject. If the mouse licks when it shouldn't, it sees this stimulus and it licks. It's called the false alarm. Many of these ideas come from signal detection theory. There was a time when operators were transmitting information by hitting on a button for a dot and a dash. And there was a whole theory and a whole mathematical description of was it a dash or was it a dash? And out of that came the core of transcontinental transmission. We use that same mathematics to understand whether a mouse's brain is able to sort this kind of task properly. And if the mouse does not lick when it should, that's called a miss. So here's what it looks like. Okay, I hope I can get this out. Okay, so here's the mouse. Here's the target stimulus. Then the mouse licks. The spout comes forward. Okay. Now, here's a non-target stimulus. The spout comes forward and the mouse does not lick. And here's a target stimulus. And the mouse licks. Okay? So this is a highly trained mouse. It's amazing. But then we play another trick on the mouse. So far it's a decision task. That's a component of cognition. We have studied this. But for today, just with one slide or two slides, I'm going to make the point of an internal state of attention, because that's what I'm telling you about. In certain blocks, we don't bring the spout forward. So we present the appropriate stimulus, but we don't bring the spout forward. So if you do this even one time, the mouse knows these guys are playing a game with me. They are showing me the stimulus, but they are not giving me anything to lick at. And they, of course, we give the mouse a little bit of chocolate water. And the mouse has been deprived of 30% of its water so that it is motivated to lick. So when this happens, the mouse switches off, because then it pays no attention to the stimulus, even though the stimulus is going on and there's one stimulus or the other and so forth. And that's what this is about. So either the spout comes forward in the engaged trials, or in the passive trials there is no spout, but the visual stimulus is the same. And during the passive trials the mouse cannot lick. So that's what the P means, and that's what the lick means. Is the mouse paying attention? Yes, it is. Because we can monitor the mouse's pupil by putting a teeny tiny, highly accurate camera. Remember, a mouse is only 15 grams, it's just this big. The eyes are just 2 millimeters big, but we can put a camera on that eye, and we can track the mouse's pupil as an indicator of whether it's paying attention. And when we pay attention, our pupils dilate. And so we know that the mouse is paying attention during engaged trials, and it's not during the passive trials. So what does this? And then during all this time, we are recording the activity of brain cells. And our technologies have advanced such that we can make genetically encoded calcium indicators be expressed in the mouse's brain, and we record from the hundreds of neurons during the same task, whether the mouse is when the mouse is paying attention or when it's not. It's a humble but extremely accurate way of monitoring the mouse's model of the world and asking what happens to the brain. And when we do this, we record from the primary visual cortex and we record from the parietal cortex. the same area that exists in humans. And here's what we find. In the primary visual cortex, when you show, remember, these are neurons that are orientation selective. So here's a neuron that likes horizontal orientations, and when this orientation comes on, this neuron fires. When the vertical orientation comes on, the neuron does not fire because it doesn't like that orientation, and then the neuron fires, and all these pink bars, I hope you can see them, are when this neuron is firing. Here are the parietal neurons, the parietal neuron, and it's firing to the same orientation because it is in the engaged condition. You remove the lick spout, the same stimuli come on, the visual cortex neurons keep firing, but the parietal cortex neurons are completely silent. So that's what this means, engaged versus passive. During the passive condition is the same visual stimulus. I remind you, the visual stimulus is identical during the engaged and passive trials. Only the internal state of attention is different. That internal state modulates the parietal cortex neuron such that it's completely silent. And this is what I mean by a neural correlative cognition. It's as if you're sitting in a class and you have switched off. Some of you may have switched off already. You're saying, I don't know what the hell this guy is saying, and you are daydreaming or you think of nothing else. I assure you your brain is flat, because that's what lack of attention does. One more twist. This is an internal representation of space, or of a stimulus in space, in which this area is very important. What about an internal representation of time? And we have internal representation of time as well. And we can do that very accurately as well in this kind of task by adding a delay between the stimulus and the response. You show a stimulus and then you wait for a while, 3 seconds or 6 seconds, which is a lifetime for a mouse. And then you bring the lick spout forward. And the mouse should lick when it sees the correct stimulus. And if it licks to the incorrect stimulus, we give the mouse a little bit of quinine to discourage that. And so yet, if it licks, we immediately know that that's a mistake. So here's what it looks like. And so then, here's the mouse. Here's the target stimulus. There's a delay, 6 seconds of delay. And then the spout comes forward and the mouse licks. If you give me a phone number and you ask me 6 seconds later, what was it? I'm not sure I'll remember. So it's a very long delay for a mouse. And in this mouse, then, we are imaging, we are recording the activity of the first stage of the cortex, the next stage, and another stage. So V1, the parietal cortex, and the frontal cortex. This is the highest stage, is also the motor cortex in a mouse. And here's what we find. That it's in the motor cortex. These neurons, so these are hundreds of neurons, and the delay is 6 seconds. Here the population response is in the visual, in the parietal, and in the motor cortex. And in the motor cortex, this delay period activity shown here is prominent. In the visual cortex, there is no delay activity. There is a stimulus, the neurons fire, and then the neurons stop firing. In the parietal cortex, a few neurons continue to fire. But in the motor cortex, a lot of neurons are firing. And this is an internal representation of time. Because I remind you, during this delay period, there used to be a stimulus, and now there is nothing in front of the animal. That is just the animal's imagination, that I must lick as soon as I can. And that imagination has this neural correlate in the activity of these very high-level neurons in which this internal representation of time is stored, and the mouse then licks when it does. So these are two simple ideas whereby we can demonstrate for you that the mind has its basis in the brain, and that internal models that make the mind in space and time have their basis in the firing of neurons in specific higher areas that receive input from lower areas, and this activity is generated within these areas. How can this activity be generated? Here is a little bit of math. And this again, I want to emphasize all of the things that you have heard from Partho today or from others, how important interdisciplinary work is. I'm an electrical engineer. I've become a biologist, and I use use mathematics all the time, and I can do computer programming as well. And so, and all my students can. And so here's a little bit of understanding, a mathematical understanding of the neural dynamics, or a mathematical understanding of mental activity. We can ask, what is the nature of the activity in the motor cortex that creates, that underlies this internal representation of time? If you have 2 neurons or 3 neurons in the brain, and they are firing, then this 3-dimensional space of these neurons represents the coding by these neurons. If you have 100 neurons, that's a 100-dimensional space. If you have 1,000 neurons, that's a 1,000-dimensional space. These are the kinds of questions we can ask. What is the nature of information? And we can do what's called a dimension reduction. From 100 down to 2 or 3 or 4, how many dimensions represent most of this information? That's called the principal components analysis. And I'll just show you what this dimension reduction analysis does to the motor cortex. In the motor cortex, essentially there are 2 dimensions. One dimension is a bunch of neurons that represent the stimulus and the delay. And another set of neurons, so that's the stimulus and the delay. And another set of neurons represent the activity. What does this mean? In any complex system that has many moving parts, such as in an airplane or such as in a complex machine, there are states of the system that lead to stable performance of the system. These are called attractor dynamics. Machines have them, and the brain has them too. And these attractor dynamics are such systems, such complex systems, nonetheless have stable mathematical descriptions that underlie the components of the mind. And finally, the mouse sometimes makes mistakes. I told you that it sometimes licks when it shouldn't. That's a false alarm. Or it doesn't lick when it should. And that's a miss. Why does the mouse make a mistake? It's because its brain makes a mistake. And you can see that when the mouse licks when it shouldn't, that's a false alarm. There is enhanced activity in the frontal and parietal cortex. Or when the mouse does not lick when it should, there is reduced activity. The next question to ask is, why is this so? And that's a very deep question. That has to do with imaging the whole brain one day, which we will do one day because of new technologies that we and others are developing as part of the US Brain Initiative. So then, we started with 2 operational questions. How is the brain wired? The brain wired with self-using mechanisms of plasticity and specificity. How does brain wiring create cognition? Neural dynamics and distributed hierarchical brain regions represent internal models, internal representations of space and time. And that leads us, in the last 5 minutes, to ask, how might this knowledge help us understand and treat brain disorders and diseases? And how might this lead to a glimpse of the next generation of computing machines? So first, brain disorders and diseases. This is a picture from the World Health Organization that shows the disease burden of brain disorders. And brain disorders are expected to overtake heart disease and cancer as the most debilitating of human conditions. You heard from Mammon about how cancer, we are developing such interesting new technologies such that if I have one kind of cancer, I want Mammon to be my physician, to take a biopsy, to do his genetic screen and tell me what the mutation is and what is a drug that can target the mutation or the product of the mutation such that my cells get killed and they don't continue to divide and create cancer. Okay? Heart disease similarly has become increasingly, at least in the richer countries of the world, a chronic condition. So if heart disease and cancer become treatable, what shall we die of? What shall we get burdened by? Brain disorders. As the population ages, the US Center for Disease Control estimates that 50% of the people over the age of 85 will likely have Alzheimer's disease. For those of you who haven't heard of Alzheimer's, I don't think there's any such person. It's a devastating disorder of loss of memory, loss of self. But then there are the neuropsychiatric disorders. 10% of people suffer from neuropsychiatric and neurological disorders. And that's the disease burden shown in bright red in many of these countries. India is an orange. And many people think it could be much redder. because brain disorders are a source of shame for many families. Yet brain disorders are ubiquitous in any society. And we must understand that brain disorders are not the fault of the person just because the person has a disorder in something we cannot see. The person has a disorder of cognition, but that cognition is made possible by the brain, and there's the disorder of brain function. So the worldwide rates of neuropsychiatric disorders, such as autism, schizophrenia, bipolar, and other things, and of course, the major unmet need of treatment that target these disorders. There is not one mechanism-based treatment for a brain disorder. At best, there is a treatment of L-DOPA, or giving a dopamine precursor for Parkinson's, but a large number of people with Parkinson's don't take that medicine because when you take it, it shuts down whatever little dopamine that your brain makes. So there is not one mechanism-based treatment for a brain disorder, not one. All the treatments that you get are treatments of symptoms. It's as if you have malaria and you go to a doctor and the doctor gives you aspirin for your fever. It does not treat the balance. Alzheimer's is devastating. There are brain scans for people with Alzheimer's showing reduced activation and increased neuronal degeneration. But then the neuropsychiatric disorders, and I run a center whose goal is to understand fundamental neuroscience and use it to begin to understand the disorders of social cognition, such as autism. Autism is a very difficult disorder because it strikes children and is marked by severe and sustained impairment in social communication, by restricted patterns of behaviors and interests. It's obviously a disorder of the social brain. It's diagnosed quite early, and parents suffer just as much as the children do, because a typical comment by a parent is, my child ignores me, or my child cannot speak to me, or my child is in his or her own world and is 4-to-1 boys over girls. My own laboratory studies a subset of autism called Rett syndrome. Just a couple of slides, I'll give you a little glimpse of how the understanding of the brain and of cognition gives us a window into understanding Rett syndrome. It's a devastating disorder. It accounts for a few percentage of mental retardation in girls. It's caused by a single gene called MECP2, and it has many different symptoms, all of which lead to very severe loss of function, such as loss of language, loss of movement, a very severe handwriting. To understand a disorder like autism, not only do we need to know the gene, but we need to make models. And we make models by making mice in which this gene is mutated. And we apply all of our knowledge of brain plasticity and dynamics. And out of that has come the understanding that in these mice, synapses are hyperplastic. And out of that has come the understanding of a molecular pathway and even a therapeutic that might make these synapses less plastic and hence more normal. And I just wanted to include one slide from a Presidency graduate, Abhishek Banerjee, who was a postdoc fellow in my lab. And he did this beautiful experiment using genome editing technology that we have been discussing. It's the latest technology for removing a snippet of a genome, in this case, one gene, the MECP2 gene, from neurons in the brain. I'm not going to go into the details, but it shows what Abhishek found in a paper that was published a couple of years ago, that in normal mice, the orientation tuning of neurons that I've been telling you about is very sharp. But in these neurons in which this gene is edited, single neurons in which this gene is edited out, the tuning is broader. The tuning never sharpens in the way of activity-dependent plasticity in a normal mouse. And the tuning is broader so that there is reduced selectivity and the responses are less. And then we and others have used human neurons. We can make neurons from skin cells in the manner that Dolly was made, by taking these neurons but then using the genomic information in a skin cell to transform it into a pluripotent stem cell and then make neurons out of it. So if I have autism, I hope one of my students will do that for me, and in a dish make the neurons that has my genetics and ask, what can these neurons not do? And what these neurons cannot do is that they don't make action potentials and they don't make synapses very well. And all of that leads to the idea that there is a potential therapeutic based on this fundamental neuroscience that is now in clinical trials. And finally, I'm going to stop in 2 minutes. Let me speculate a little bit on computation and machine learning. How many of you have heard about AlphaGo? Oh, good. How many of you have heard of Lisador? Well, good. So at least I'm delighted there are some young people left in the audience and were not taken out to see a movie or something. Okay. So earlier this year, a computer based on machine learning beat the world Go champion, Lisador, 4 games to 1. Go is a game of strategy, just like chess. But it's widely acknowledged Go is one of the most difficult games of strategy in the world. Chess is a game of capturing one king. Go is a territorial game. It's of capturing more territory on the board. Both require strategy. In 1997, a computer program based on machine learning and artificial neural network beat Garry Kasparov in a 6-game match. It has taken 20 years so it did its program. The engines, the machine learning engines driven by Google or by Facebook, need billions upon billions. They are built on billions and billions of users and programs. Google has 1.5 billion searches a day. Facebook has 1.8 billion active monthly users. India itself has 142 billion users. So you need Facebook or Google to supposedly stay in touch or do your searches. But Facebook and Google need you too. They need every hit. They keep track of every hit that you have made. Every click. And they store it away. They store it against your computer. They know where you are. They know what you are searching for. And they use it to refine their algorithms. Because from these millions of examples come the weight that shape the transformation from the input to the output. This is a deep disconnect between how a machine learning program works and how humans learn. I can take a 2-year-old child and I can show that child 2 dogs or 2 pictures of dogs, a small dog such as a bulldog and a big dog such as a German shepherd. And then I ask her, then I show her let's say a greyhound or a midnight dog. What is this? And she'll tell me without hesitation, this is the dog. From 2 examples, a human being learns. Whereas this would take impossible numbers, thousands upon thousands of examples for a computer to learn. So where does that take us? Well, I think that the mind works in somewhat similar ways to how a computer works, in the sense that machine learning algorithms also build a model of the world, but they implement that in very different ways. I've told you that the way that the brain is built relies on specificity and plasticity, which create circuits, which create dynamics, which form the algorithms for building an internal model of the world or the mind. Might machines be built that way? They might, and there is certainly a need. This is my last slide. There are many issues with computers today. Computers have very large power consumption. The AlphaGo computer needed 300 kilowatts of power. The human brain works on 20 watts of power. There are many other issues. The computers have a central power unit. They have a fixed architecture, whereas brains, I've been telling you this, has flexible, reconfigurable architecture based on plasticity and learning. And so on. And importantly, the power of modern machine learning, which is what makes a lot of things possible. That's when you call Uber or when you do anything that involves a computer today, it has been built on the ability to cram more and more and more and more memory and processing in chips. That would end in. The brain does not work that way. And of course, the goal of computers is computation. The goal of brains is computation. What might we learn from brains that can be used for machines? Well, human beings were fascinated by flight. They could see that flight was possible when they looked at birds. But it took human beings the conceptual leap to understand that flight is not about the structure of feathers or the flapping of wings. Flight is about countering weight by lift and by countering drag with thrust. And when we understood that, then we built machines that use the same principles that birds do, but at very different form and architecture. And I believe it will be the same with understanding how neuroscience can be used. But where does that take us? And I want to end by a little bit of thinking about what does all this mean for society. So I believe that along with the big questions of our time, such as climate, such as energy, the understanding of the brain and mind and its impact on health and on computation, on machine intelligence, and in fact, on the nature of work itself, because with machine intelligence comes automation. It will be the central transformations in the next few decades, certainly in your lifetime. So should it happen? We asked, should gene editing be done? And we heard from Balu about the moratoriums that people put. I believe it will happen. Human beings keep pushing forward things, sometimes in unthinking ways. But the immediate question to ask is, who would benefit? How would it impact society and you as its future leaders? So an important question for every society to ask and answer is, how are the fruits of its science and technology distributed? Who has access to them? We heard from Amin about access to healthcare that, say, a society like Cuba provides. And we know about the digital divide in this country. So I believe that these advances, useful for treating our most difficult conditions such as brain disorders and for building possibly the next generation of computers, they have the potential to improve life for all citizens. But the relationship between technology and society is very complex. Technology shapes society. Look at how much our cell phone has influenced us, or our computers influenced us. But then society shapes technology too. And therefore, alongside these technological transformations, I hope we will also join in the social transformations that will make these advances available to all. Thank you very much.
Yes.
I have two questions to ask. First,
Yeah, go ahead.
we know that our brain is dynamic and
Wait, raise your hand. I thought you were somewhere else. Please raise your hand so that I know where you are. Yes. Okay, sorry, yeah.
we know that our brain is dynamic.
Uh-huh.
Standing in a particular time, we want to concentrate on a particular part. So, in a particular time, lots of thoughts are coming in our mind or as well as brains. So my question is, what are the orientation of brain cells or neuron reactions happen in the brain when we try to concentrate?
So concentration is a form of attention. Okay? And in fact, our brains work best when we are attending. It is a myth that we can parallel process or that we can do a lot of things such as driving and texting and looking at a video all at the same time. So we do attend, we do concentrate, and concentration or attention is a very central part of how our brains work, and we should acknowledge that. I don't know if that answers your question, but that's how I see it.
So firstly, thank you for your very lucid and informative talk. I'm from the Department of Sociology, and I have a question which we often try to tackle philosophically. For a congenitally blind person whose visual cortex has never received a visual impulse, if he gains the capacity of sight or capacity of vision, can he comprehend what he sees?
Yes, that's a very good question. My friend Pavan Sinha from MIT has been in a mission, and I know that Balu and L.V. Prasada Institute, as well as the Shankar Netralaya, many places in India have been doing remarkable work removing cataract, doing cataract surgery in older people. So India has the highest incidence of congenital cataract in the world, and this is a preventable situation, and it's worsened by the lack of access to affordable treatment. So there is a population of people whose cataract has been removed and replaced with an artificial lens at a later point in life. And you can ask, how do they see? What do they see? That's the closest we can come to your kind of question. And the answer is that they can learn to see. But whether they will ever see as well as if they had vision from childhood or from birth remains to be seen. But they do certain things they learn, and it has turned on its head a little bit about our ideas of vision. For instance, when something moves, we used to think that we first identify the object and then we track its motion through space. But it turns out that when an object moves, it becomes more visible because the different parts of the object can then cohere. And so computationally and certainly perceptually, movement and hence identifying objects by movement is one of the first things that recovers.
Sir, as you told that a cognitively blind person's visual cortex is active, so we generally have this idea that cognitively blind person, they are sensitive to auditory reflexes and to touch. So those those visual cortex, do they contribute to that extra sensitivity?
Yes, I mean, congenitally blind individuals, because they have retinitis pigmentosa or whatever, due to whatever reason, if they have no transmission of information from the eye to the brain, the visual cortex, and a large area of the cortex, therefore, is sitting there without any visual input. And the brain rewires itself, either directly or indirectly. Neurons are connected across cortical regions via multiple synapses. So auditory or somatic information enters the visual cortex. And I gave you an example of how auditory information comes to be processed in the visual cortex. Does it enhance auditory function? The answer is yes. In fact, interestingly, if you have blind people who learn Braille, then as they learn Braille, the sense of touch takes over the visual cortex and replaces sometimes the sense of hearing, which had been there and stuff like that. There's all this dynamics of plasticity that goes on. But indeed, new tasks and new competences are mediated by new cortical areas made available.
Do we always approximate continuity by discreteness, as you talk about cells, and is that how we see continuity? And my second question is, you talked about homologous from mouse breed that are homologous to the humans. If you look at the genes that express for a specific stimulus in the mouse, do we also get the homologous genes in the human responding in the same way?
Very good questions. So there are neurons in the retina that respond to motion. So discrete pixel by pixel computation of motion is not the only way. There are local motion detectors. But the global detection of motion, if something moves across the visual field, there is no neuron in the eye that can detect that. It has to be detected by either a serial set of motion detecting neurons in the retina that then get linked up through the visual pathway to other neurons that have a much more broader view of the visual field, or most likely, and we know this from various experimental results, there is a combination of things that provides input of that information about such global motion. But very local motion can be detected in the retina itself. And on the business of the homologous genes between humans and mice, there are many, many genes that are homologous, but we don't know which genes are used to make the parietal cortex yet. There's a lot of gene expression studies being done, but it's a very complex combinatorial question. How does the cortex get to different areas is an unsolved question yet. Yeah.
Sir, my question is this, is it possible to understand the activity of brain with the help of quantum mechanics?
Well, there have been some attempts, and the answer is I don't know, but I'll give you a very broad answer, that in science there are levels and levels and levels and levels of analysis, but in general it's very difficult to bridge across too many levels of analysis. Okay, this table is hard. It is difficult to explain. I mean, we can seek an explanation for this at the level of its molecular structure. But then going beyond that, going into the elementary particles and the forces in the atoms that hold the atom together, that's not proven to be a very useful level of description for a higher level of observation. So that's just my view of where should we seek explanations. And if the appropriate level of description, as I believe, is the mesoscale level, at the level of single neurons and their firing, how that encodes information and how networks of neurons then shape and read out that information, then going one step below to how do inputs and synapses help shape that is useful. Maybe one more step, how do the molecules inside synapses help shape the synaptic strengths that allow the neuron to weight its inputs, but then beyond that, I'm a little bit skeptical.